Hello, everyone, and welcome to our webinar discussing causal AI in precision medicine. Thank you for carving out time during your days to join us. My name is Ashlyn Alisi, and I am the marketing director here at BioAI. Today, we will be discussing genomic sequencing, computational processing, and groundbreaking AI strategies that are reshaping the landscape of healthcare and pharmaceuticals, as well as how these new technologies can help in developing more efficacious medicines and personalized treatment modalities for better patient care. We'll have time for questions at the end of the presentation if you would like to submit any in the chat on the right here. And before we begin, I do want to give a quick introduction to our key speaker today, Dr. Tom Chittenden. He is a Giga Society Fellow with a history of over 25 years in experimental and theoretical research. He is the chairman of the Scientific Advisory Board here at BioAI, and he also currently serves as an honorary professor in the Digital Environment Research Institute at Queen Mary University of London. And with that, I'll hand it over to Dr. Chittenden to lead the presentation for us today.
Well, thank you, Ashlyn, for the warm introduction. I hope I don't disappoint after that. And I also like to thank you for setting up the webinar. It's going to give us an opportunity to talk about what we've been working on from a causal AI standpoint over the last 20 years or so. I'd like to also thank everyone that's in attendance here today. I can't see facial expressions here this morning on the East Coast, so I can't tell if I'm hitting the mark or I'm putting everybody to sleep. But what I can promise is I will make this as concise and entertaining as possible. I also promise no math or statistics, so everyone here on the East Coast, we don't have to worry about putting you off your lunch here later on this afternoon. So what we have been working on for the past 20 plus years or so, and Ashlyn, I'm not able, hold on a minute, there we go. Did you do that or did I do that? Were you able to advance it or was that me?
I did it, but if you can, I'm happy to do it too. Just let me know.
So we have built, over the last 20 years, we have built and continue to build biologically relevant and experimentally validated AI technology. And what do I mean by that? It's biologically relevant. So we are answering questions in the healthcare space, and specifically drug target discovery and development. Experimentally validated, we've worked with some of the top minds in their respective fields around the world to publish this technology in top tier scientific journals such as Nature, Science, and Cell. And what we have been working on very hard and heavy over the last decade or so is to integrate something called causal inference into all of our generative AI and ensemble supervised machine learning. These capabilities are capable, or they sit on top of multimodal data, which I'll explain a little bit later. And they are inherently white box in nature. So what do I mean by that? They're explainable. A biologist can understand the output of what the AI is actually telling us. And from a causal standpoint, we've gone to great lengths to integrate causal inference into everything that we are doing. And we now have scientific evidence that these approaches are capable of deriving causal dependency structures that are reflective of the signal transduction cascades that drive cellular behavior and dictate phenotype. And from my point of view, that is the single most important aspect of analyzing this data. We want to move away from what correlates with disease to actually deriving hypotheses about what's driving disease. And that is what I'm going to share with you here today. So I'm back in the driver's seat here, Ashlyn. I was able to advance it. So the multimodal causal AI, we've developed an approach called in silico phenotype projection, which is capable of integrating spatial transcriptomics and single cell RNA-seq data with causal AI models to precisely identify therapeutically relevant molecular signals in regions of interest in heterogeneous tumor tissue. Now, I'm going to take that a step further. The field used to believe that it was tumor tissue that had the biggest heterogeneity, the confounding aspects of multiple cell types driving disease. And what we're finding is there are clonal populations of cells that are driving disease in a number of different therapeutic areas. And so what everyone at BioAI is working on is digital pathology. So can you build these tools to build predictive algorithms off H&E or histology slides for known biomarkers? And the bottom line is yes, and it's very effective at doing that. The other aspect of this is identification of novel biomarkers or novel biological targets for drug discovery and drug development. And we have, again, a lot of scientific evidence showing that this is actually possible. So all of this work has been built with a team of renowned AI data scientists, software engineers, and translational scientists and board certified pathologists. And not only can we focus in oncology, but I like to refer to these tools as disease agnostic. So we're working in a wide range of therapeutic areas to address these questions. So what you see here is this continuum from discovery research tools all the way through commercial applications. And these are the applications that we have been able to develop with this AI technology. And what I'm going to share with you today is a bit of our spatial transcriptomics approaches and how we can apply that to multimodal data. This leads to drug target discovery. And then what do you do after you have a causal drug target? You're working with an organization that has developed an investigational new drug or an IND. We can help put a molecular mechanism around that IND. And then that speaks to a number of different applications, such as drug repurposing or repositioning, drug efficacy assessment in clinical trials, and a whole new area that the field is working on, digital twins for in silico clinical trials. And then the commercial applications that BioAI is specifically focused on is digital pathology and medical imaging analysis. And this speaks directly to not only diagnostic tests, but being able to screen patients, inclusion-exclusion criteria for clinical trials as well. And so what you see here with this figure is a flywheel. At the center or the hub of this flywheel is causal AI and something called scientific machine learning. And each spoke or arm of this flywheel, if you will, are the areas that we have actually developed. And I'm going to talk to you about in silico phenotype projection, the power of that for uncovering drug targets. We'll talk a little bit about the biomedical imaging and digital pathology and how we are integrating spatial transcriptomics with it, how we apply natural language processing, generative AI, and knowledge graphs to place these signals that are non-functionally annotated. The vast majority of the human genome, we don't know what it does. So when we come across the signal that is non-functionally annotated, how do we place it within known biological context? And that's how we're using our generative AI approaches. A range of disease-related questions that we can address, from liver disease, Alzheimer's, oncology, lupus, COVID-19, cardiovascular disease. I'm going to focus a little bit on that. And then rare diseases. So with the advent of single cell and spatial transcriptomics, we can now derive or capture enough biological variation within a single sample to do a lot of very sophisticated machine learning techniques, or apply a number of techniques here to capture that biological variation, to find intrabiological signals. And then we can take an old school bioinformatics and biostatistics standpoint or approach to looking at it across populations. Each turn of this flywheel generates novel insights and true discovery. So we've been able to figure out an approach to capture all those or unannotated biological signals and advance our understanding of these signal transduction cascades. It's an accelerator, if you will. And down here at the bottom, we've got the opportunity to fail early and fail fast. And I know that sounds a bit strange to a lot of the translational folks here watching today, but it really is a real shot in the arm for us to be able to tell if we have a signal in the data before moving forward. So I'm going to focus the first part of the talk here on a couple of these case studies with cardiovascular disease, and specifically in silico phenotype projection here. And this is a very busy slide, but I think that it speaks to the complexity of what we are able to capture and address to move downstream. And this was a project that we were working with Mike Simon's laboratory at the Cardiovascular Research Center at the Yale Medical School. And Mike had a question that it looked like his data was pointing to a specific signal transduction network, but it was absolute heresy at the time. He was proposing that two signaling proteins, ERK1/2 in the upper left-hand corner here, were downstream of a signal transduction cascade called TGF-beta. Absolute heresy at the time. They were firmly implanted in the classical MAP kinase signaling pathway, but all of his data was pointing in that direction. So he asked the team at the time if we could build an AI strategy to address this question. And this is what you see here on this slide. We started with human cells, and these are HUVEC cells, human umbilical cord endothelial cells. And he perturbed ERK1/2 in vitro. We extracted the RNA from these cells. We put it through our ensemble machine learning approach, where we got down to the point where you see inset 7 here, Roman numeral 7, is that we were able to derive a causal dependency structure with all of our Bayesian belief network strategies. And then he was able to validate that in vitro. So what he did was he perturbed or overexpressed this yellow node, and we knew that the network was connected because the entire network and all of these green nodes at the bottom changed state. This signal or this network together was pointing to or predictive of 3 vascular phenotypes. And then when Mike built the subsequent mouse model that was a conditional ERK2 knockout on a ERK1 negative background. So he let these animals come up into adolescence, and then he shut off both of those signals. These animals, all of these animals, presented with these same 3 vascular phenotypes. So then we were able to put it or use our natural language processing tool at the time. And it showed the biological relevance of all the unannotated signals that we were bringing to the table. So we started with human cells, and then Mike was able to create an experimentally validated mouse model that then set the stage for all of our single cell and spatial transcriptomics work. And closer inspection of this causal dependency structure, or more correctly, I'm going to refer to this as a putative conditional dependency structure, is that this is what it looked like. And Mike Simons was right. We were able to identify TGF-beta 2 as one of the driver nodes of this network. So let me explain this a little bit. On the left-hand side, we have these signals or these genes that associate with the greatest degree of conditional dependency in the model. And as you move across the DAG, or from left to right as you're looking at it, there is a flow of conditional dependency until you get down to the response genes here on the right-hand side. So if you were to change the state of TGF-beta, as we were proposing that he would do, he changed the state of the network. These response genes lit up like a Christmas tree, and basically all arrows point to a gene called endothelin 1, which was a known driver of these three vascular phenotypes. But at the end of the day, all arrows point to a gene of unknown function. And when you look at this Circos plot onto the right-hand side here, the outer blue band represents known functional relevance. And this is a very robust finding, is that we find this with any experimental design is anywhere from about 10 to 25% of the overall signal are genes of unknown function, or signals, vectors, if you will, of unknown function. And so we were able then to place all of these signals within known biological context. And just to take it a step further, we ask our friends at BioRelate, which is a biotechnology organization in the UK, to actually apply their large language models to this problem as well. So we gave them the actual driver genes here, which one of them was TGF beta 2. And as you can see, you have very, very specific routes to these three vascular phenotypes. So we were predicting hypertension, they were predicting renal function, hemorrhage, cerebral hemorrhage, and then end-stage renal function as well. We were also predicting hypertension here, but not from a renal standpoint. All of these paths, if you will, are very specific for the known biological pathways that they were able to extract from this analysis. And so we firmly implanted this 10% of the overall signal that we found within these known pathways as well. And we are still working on that. This set the stage for all of our single cell and spatial transcriptomics approaches and initiatives, which I don't have time to share with you today. So I'm going to change gears just a little bit here. And I'm going to talk about oncology for the rest of the talk, and specifically what we have done with quantum machine learning. And there was a real reason for that, is that this slide represents another very busy slide here, so I apologize, but this was one of our very first ensemble computational intelligence strategies, where we were working with the Cancer Genome Atlas data, TCGA data. It was multi-omics, not multimodal. So we had the expression of mRNA and microRNAs. We were working with something that we called somatic tumor variants, so single nucleotide changes. We had structural changes that we were looking at as far as copy number variation, and then DNA methylation. We integrated all of these platforms together. We put it through our ensemble machine learning approaches. So I've never trusted a single run of a single algorithm on a single partition of data. So we are running as many as 8 classes of algorithms on as many as 1,000 partitions of data. And we got to the point where we took 2 different strategies to reduce the dimensionality: a data-driven approach, which I'm not going to go into with any detail, but just to set the stage with what we ran into as a real problem and why we've been looking at quantum machine learning. The second approach was something that I had worked on, or an algorithm that I had developed with a number of teams over the years, called NGO-seq, where we're collapsing all of these signals down onto biological pathways. The problem with this is non-functional or non-functionally annotated signals completely fall out of your model. But at the end of the day, we got to the point where we had a classification performance of over 99% accuracy. So across 8,000 tumors, we were only misfiring on 8 tumors in that test set. The problem came with this is that we were working with 22 of the 33 cancer types. So we weren't doing very well from this multimodal or multi-class approach with this strategy here. And what we found is that those 11 tumor types in TCGA were below about 100 samples. And so that's when we started looking towards other options for statistical optimization that could help us address this. And so this slide, what you're looking at here, was the first successful demonstration of a multi-omics cancer patient data approach with quantum machine learning. And specifically, we had a very, very specific and difficult classification program or project here that we were working on. We were trying to delineate luminal A and luminal B breast cancers. We had 311 tumors that we were working with. And then you can see the breakdown on how we were testing and training. And so on the right-hand aspect of this figure, we have 250 patient samples. All of the algorithms except two of the conventional algorithms, which is, I believe, and I'll have to go back in and look on this, but I believe it was naive Bayes and random forest were not working very well with this classification program or project. But as we were systematically decreasing the amount of the fraction of the training data, as you can see here, when we got down to about 45 patients, all the established algorithms completely fell over. And what we were left with at the end of the day was quantum machine learning. And this was an approach here that we had taken. It ended up landing on the cover of Patterns, so it's a Cell Press journal. But this was quantum annealing, and it took a very long time for it to get published. And I think it's very important to be transparent with all of this work, is that it worked. We showed the first demonstration of successful classification with multi-omics data in a basically on clinical samples here. But what was also very informative or efficacious here was our simulated approaches. So simulated quantum annealing was doing very well also on this. And so it took us a while to convince the reviewers that quantum annealing is actually here. The technology as it is today is very efficacious. And this has set the stage for a number of different initiatives that we're working on with the government now to advance quantum machine learning in drug discovery and development. So now this takes me to what we have done specifically with BioAI over the last year and a half or so. And we were focusing on non-small cell lung cancer and how we were integrating causal AI with spatial transcriptomics. And this is a very exciting initiative here. And so I need to set the stage a little bit. What you have on the left-hand side is a 10x spatial transcriptomics image here, and that you have 6,000 spots across this tissue sample, all representing the entire transcriptome here. And I'll explain a little bit more about the experimental design, but we were trying to differentiate between normal tissue, which is highlighted in orange, versus the blue, which is the tumor tissue as well. What I had shared with you in the cardiovascular space is over here on the left-hand side, these directed gene expression networks. They're all directed, so there are arrows on these edges. But what we were able to do or to develop was a directed tissue spot network, which is very exciting here, is that ultimately what this is telling us is that we can pinpoint down to a 55 micron region of a tissue where we believe this aberrant directed gene expression network is originating from. And we believe that this is going to advance drug target discovery, as well as being able to predict novel biomarkers in any given therapeutic area as well. So this is what the actual directed spot network looks like. And again, the flow of conditional dependency is from left to right. These yellow spots represent driver spots, and these green spots actually represent response spots. We had a subnetwork here that I'm going to point out when we look at the expression profiles of all these signals here, but we had a subnetwork here that was not originating in the same location where the other 5 spots were actually originating from. And so we have inverted here, or now it's from a vertical standpoint, the same tissue section or sample here. These were all the spots that were annotated by Mike Bonham, our pathologist. And as you can see here, the yellow spots are originating here with the border, or right on the invasive margin between the normal tissue, the stroma, and the actual tumor tissue itself. We annotated, or Mike annotated, a number of spots within the tissue as well. And what we're finding is that, one of the 6 signals was originating within the tumor. So it's much, much more complicated or complex than we had originally thought. But when we start looking at this, and this is Mike's interpretation of this tissue sample here, is that there is a wavy morphology associated with this invasive margin right here on the border of the tumor in blue, and kind of the grayed out area here with the stroma. So right here on that border is the invasive margin, and then immediately adjacent to that is the normal stroma. And we were able to differentiate these causal signals that were originating on that tissue. And so this is what the actual molecular DAG looks like here. And again, it's tipped over onto its left side for everyone that's familiar with these directed acyclic graphs. You can't see the arrows except on the response genes here, but all of these edges have arrows on it. So the flow of putative conditional dependency is from left to right. And I don't want to leave anyone unsatisfied here, but we have de-identified all of these signals till we get a little bit further down the road. This is a lung neuroendocrine tumor. This is a rare disease state. And what we are finding is I put the molecular functions around the drivers and the response genes to put a little bit more color around it. And we had a positive hit. So Mersana Therapeutics here in Cambridge, Massachusetts, has an IND that is in phase 2 clinical study for non-small cell lung cancer. And it was pretty high up within the DAG here. I would say that they got it 90% right here, and they're looking at ovarian cancer as well as lung cancer. But we found a signal that is very consistent. This aspartic endopeptidase family is actually driving the state of this IND. And when you look at the signaling or the functions of all of these genes on the right-hand side, you see some major signaling networks here. The VEGF-R2 signaling pathway and PI3 kinase AKT signaling. These are all signals, or these are genes that are responsible for driving tumorigenesis and lung cancer. So we are finding signals well upstream of all of known signaling pathways. And then we are able to isolate that signal down to about a 55 micron region or spot within a tumor tissue sample. So as far as how informative these 600 genes are that we actually identified within this approach, they're highly informative. So these are ROC curves here, and this represents 100 different partitions of that data. So 100 different runs with 2 different algorithms, so XGBoost and Ridge, which is a penalized regression approach. You can see that there's near perfect classification at the molecular level between being able to predict tumor versus stroma spots here on this tissue. And so when you look at the expression profiles of the driver genes themselves, you can see that all of them are isolated to this invasive margin region here, right on the border between the actual tumor and the stroma. This gamma binding protein was the one that we found that signal was originating within the tumor itself. It looks like it's more evenly expressed across the entire tissue here. So we look at this as a sub-network. But then when I started looking at the actual pathways from this endopeptidase, which is that driver gene, and it's also expressed in lung adenocarcinoma, which I'll talk to in a minute, is that when you delve deeper down into the DAG until you get to this histocompatibility complex protein, which is one of the response genes, you can see how the expression changes throughout the tissue. And what I want to bring your attention to is this glycoprotein in this collagen type 3 protein. These are major hubs, not only in neuroendocrine tumor that we identified it, but also in lung squamous and lung adenocarcinoma signals that we were looking at as well. When you get down into the bulk of the actual network itself, we found 2 proteins that are specific to neuroendocrine tumors. This PDK signal is an ortholog of PDK1, so another positive hit. And then we had this neuroendocrine protein that is, both of these proteins are highly expressed within the tumor, not within the invasive margin, nor within the normal tissue or the stroma as well. So the theory is, and what we have scientifically validated time and time again, is that if you turn off this signal, we would predict that you are turning off these signals that are actually driving tumorigenesis within the sample or within that patient. So now when we look at lung squamous cell carcinoma, this is the DAG here, again, tipped over onto its left side. We didn't find the exact same driver gene here within lung squamous cell, but we did find the C12 peptidase family. So basically the same molecular functions that are going on that are driving the neuroendocrine tumor are also driving lung squamous and lung adenocarcinoma samples as well. When you look at the response genes here, we have an endocrine receptor and a protein tyrosine phosphate receptor that are known drivers of lung squamous cell carcinoma. So again, a working hypothesis that now we can hand off to our experimental collaborators that then validate for us. So what this is actually showing, and I'm going to show the same thing here on the lung adenocarcinoma, is that Mersana Therapeutics was very close, and looking at this in lung cancer, we find that same endopeptidase that's actually very high up in the DAG, driving all of the signal within lung adenocarcinoma as well. If you look at this, the downstream, we have a RAS oncogene family member that is known to drive lung adenocarcinoma. We have a number of other signaling proteins that serve to drive tumorigenesis as well. So the bottom line is that we are finding shared and unique signatures from an individual sample themselves. So those unique signals that we are finding are inherent to the actual experimental design under study. But then when we can pinpoint where those signals are coming from and the families, the molecular signaling associated with it, then we can take more of a population-based approach and look at how frequent these signals are occurring in samples. And that ends up being distilled down to something that's very manageable as far as computational biology and biostatistics approaches to looking at the frequency of these signals. So we're very excited about this, and this sets the stage for a number of commercial applications that BioAI is actually going after as well. So remember what I had said here on the left-hand side of the DAG are potential drug targets. On the right-hand side are response genes or biomarkers as well. And so we have put an initiative together at BioAI here that looks at a lung screening panel. So we're looking at known biomarkers in lung cancer and being able to predict their status with H&E. So with a histology block, can we build computer vision approaches to actually predict biomarkers. The bottom line is yes. We have validated this approach, and we have a manuscript that has been submitted that is currently under review with Lilly, and it was on the RET biomarker for lung cancer. But you can see that we've got a half a dozen other markers that we're actually looking at. And here, if I can get this out of the way, we have another initiative here called STP and the structural causal modeling. And this is simulated transcriptional profiles. So can we enhance the classification performance and make these algorithms more generalizable in the long run? You know, the aspect of all this, or the non-generalizability of machine learning, has plagued the field for the last 30 years. And so the bottom line is yes, you can integrate these molecular signals with your computer vision strategies or digital pathology strategies to build more robust assessment tools. And so just giving you a bit of an example here, next couple of slides here, of what we're working on at BioAI. So how do you integrate these molecular signatures with the computer vision models? And so you're integrated pixelated, or trying to integrate pixelated vectors and concatenating them with molecular vectors from the signaling data. The short answer is yes. And what you're looking at here is the informativeness of these molecular signatures that are above and beyond most computer vision models right now. And so what you have here is a ROS positive, it's a ROS fusion positive lung adenocarcinoma tumor. This is the expression profile on this 10x tissue sample. And then down below it is a ROS negative, so without the fusion. And what you have to the right-hand corner is a box plot of a surrogate that we're using for the actual fusion itself. So with the fusion positive sample, you can see that the expression, or it's driving the expression of ROS1 on the right-hand side, versus the red signature or signal that you hear, which is absolutely, you don't have much ROS signaling or expression. So our ability to delineate tissue spots, so from the ROS1 positive versus the ROS1 negative tumor tiles or tumor spots, you get near perfect classification. However, when you get to a point where you're looking at integrating multiple biomarkers within a single strategy, this is where it gets a bit more complex. So again, the upper left-hand corner here, we've got the ROS positive fusion sample, and we've got an EGFR positive signal down below here, or sample. And as you can see, the EGFR mutation is actually driving, not to the same degree, but it is driving ROS expression. And now when we look at the classification performance here, we go from near perfect down to about 70% classification performance. And that wouldn't do much for us by integrating a molecular vector into our computer vision models here. And why this ROC looks the way it does, again, this is at least 100 partitions of that data. And what we're seeing is that they're very consistent. We're not seeing the same degree of classification performance, but you aren't getting the overfitting within the models. So this last figure here, this last slide I want to show you here, is then what we do in that same experimental design when you're looking at ROS positive versus EGF positive samples. What we simply did is we didn't integrate the actual causal DAG in this model at first. We just took the 100 most positively correlated signals or expression signals to that ROS1 fusion. And then we took a look at classifying these actual tumor spots again. And as you can see, when you take this approach, which is a little bit more sophisticated and less naive, with the random forest in the ridge L2 penalization approach, is that we're back up to near perfect classification approaches. So what we are actually working on now is being able to take multiple signatures, multiple vectors, and concatenating them with the pixelated vectors within our computer vision models. And the last slide that I want to present here is a direction that the field is going here, is the use of structural causal modeling in digital twin technology. So I can't see everyone's facial expressions, and I usually get a number of eye rolls here. There's a lot of hype, not only within the AI space, but specifically with digital twins. And there really isn't a solid definition of what a digital twin is in the drug discovery, drug development space. But what we are proposing is the world's first exascale causal AI platform to sit on top of all this probabilistic graphical network strategies that we have developed. So we are looking at thousands of disease relevant or related variables from patient cohort data. And then that will allow us to go in and engineer digital twins so that we can run virtual patient populations or create virtual patient populations and run in silico drug efficacy trials. And this speaks to drug repositioning as well. So we can work very closely with a pharmaceutical company. They may have, or we can go into their arsenal of shelved INDs and look at assigning a digital or a probability, a putative probability of how effective various therapeutics, whether they're INDs or fully baked drugs that are currently on the market, and is there an opportunity to repurpose those signals when we know a mechanism of action that I had spoken to earlier. So it's a very exciting time to be in the space. There's a lot of work that needs to go into this, but the rest of the field is moving in this direction as well. So that's all the slides I have. I've got one more for the acknowledgments here. Very important to thank everybody at BioAI for all of their help in putting what you have seen together today. Ashlyn, again, thank you for the opportunity here with the webinar. Mike Shea at IBM and Sir Mark Caulfield and Greg Slaubaugh at Queen Mary, University of London. I had mentioned Mike Simons at Yale, Daniel Leiter at the University of Southern California for all of our quantum work. Siamak Baram at the University of Strasbourg. I didn't have the time to present that data as well, but we were working very closely with him during the pandemic, and we were actually able to block SARS-CoV-2 viral uptake and replication in human lung epithelial cells, which was very exciting. And then all of our friends at BioRelate, and specifically Mark Hughes. So thank you for your time today. And I've noticed it kind of threw me off a bit there, but there are lots of questions here that I think Ash is going to have to sift through here.
Yeah, thank you so much, Dr. Chittenden. Yes, let's start the Q&A discussion. So we have a lot of questions, and what I'll do is I'll post them in the chat here so that you can see them, Tom, as we go through them. So this is the first question here. Was this sort of work able to be confirmed by removing the theorized gene, causing the intended problem, example, hypertension, and then adding it back to a knockout to recover the phenotype? I think this was in regards to slide 12.
Yeah, okay. So let me go through this because I was at the top reading the other question. Okay, so you're starting down here at the bottom. This was the first question. They came in this order.
Yeah, I have them all noted.
Confirmed by the removing the theorized gene by causing. Oh, okay. So this is very good. Ash, can you go back to the slide? No, this is a very insightful question here. It's not slide 12. Earlier. So we're off, I think, by 2 or 3. Keep going. So we're one more. we're talking about this slide right here. From a phenotypic standpoint, were you able to normalize the hypertension, or were you able to favorably normalize the phenotype within these mice? The hypertension question, yes. So that did actually stabilize here, is that when we were looking at what's driving that and what's not driving that, and what I was not able to get into from an in silico standpoint, your bottom line for that question, very insightful, is yes, it favorably modifies the phenotype that we were looking at. From an in silico standpoint, this is where we had developed something that is beyond what was presented here in this figure. This is Bayesian network structure learning. And then we have built downstream from this, and it took us a couple of years to do that, structural causal modeling, where we can actually get into counterfactual analysis and potential outcome analysis, where we can tweak these nodes of interest, and we can look from an in silico standpoint what the effect would be on phenotype before we actually even go into looking at that from an in vivo standpoint. So that was a very insightful question. I didn't have time to talk about that, but that's all baked in now to a lot of what we're doing from an in silico standpoint, and then looking at, you know, its efficacy from an in vivo standpoint, what that actually ends up doing. So that is all baked into what we're actually doing now.
Amazing. All right, next question. You mentioned the example is multiomics, not multimodal. Can you please explain the difference?
Yes. So what we have is, we used to have a slide here, and I don't believe I've got it now, but multimodal, when I refer to multimodal now, we are looking at real world data. So EHR data, and integrating various phenotypic data within the model as well. So just not looking at it from a multi-omics standpoint or looking at the molecular signatures, but how those molecular signatures are affecting various phenotypic data within the model. So Ashlyn, can you go to the last slide, please? And I don't think I've got right before the digital twin slide. So this is a representation of these probabilistic graphical networks. A lot of individuals refer to this, or in the field refer to these as knowledge graphs now, where you have phenotype data within your model. So that is what we are going after. So we've got a loose molecular or mechanism of action at the molecular level that we can tweak those nodes after we have identified them, and see how all of that propagates through the actual network itself. And so we need to build that on top of multimodal data so that we can look at the effect of phenotype. What I had shown you today, and what we had done with the counterfactual approach, which is really potential outcome approach, is we were looking at the BIC score, which is a Bayesian information criterion score of that overall network. And we were looking at how that actually changed itself. But now what we're doing is we're using that multimodal data. You've got a phenotype downstream. If you perturb a molecular node in there, or a signal that you believe is driving the network, what does that do to the phenotype? And so now we have built that with multimodal data, just not multi-omics data.
Okay, next question. Do you have any experience using the histology mark genomics approach on stem cell differentiation? Specifically interested here in identification of cells that are improperly differentiated?
Yeah, we do. Not from a histology standpoint, and not from a spatial transcriptomics standpoint, but from a very good question, but from a single cell standpoint. And I've got slides here, and this is, Ash, we were talking about this the other day. Can you go into the supplemental slides? And I've got it up here on my laptop, so I can take you to the area. Can you go to slide, I believe it's probably going to be 43. You're very close. Up, back one. Another one. So we've built variational autoencoders off a single cell data. And so what you're looking at now is not clustering patients or not clustering genes themselves, but actually clustering cells. And we don't have the time here, but I'd love to, we could take this offline if you're interested. This variational autoencoder approach is very robust and generalizable in clustering cells. And what you see down in kind of the lower left-hand aspect of this is a consensus cluster. At the end of the day, we are able to cluster these cells and stitch a neural network back onto the encoder where all of that housed information. This is a generative AI approach, so there's no supervised approach at all. What we can do is differentiate the clusters here, and then, Ash, can you go down another slide or two here, right here? What the world, McParlin did in 2018, and this was a very touted or well-touted paper with their single-cell RNA sequencing and looking at liver and differentiating the heterogeneity of the cell types within liver. What we ended up doing in this Nature Metabolism paper is that these were endothelial cells, and we were able to derive what it looks like this brain stem structure here. Some people look like it's a nuclear explosion, but down in the lower left-hand corner where we've designated 4, we were actually able to look at this and designate that cell population as what was driving the pathogenesis, which was actually atherosclerosis, and that led to reversing atherosclerosis, albeit in mice. Now, to get to your point with differentiation and adding temporal information on top of this, can you, Ash, go down another? There you go. So we took a second generative approach where we weren't clustering cells now. What you have on the x-axis are the cell clusters. And now what you have is that we clustered the data, and now you have the individual gene sets. And so when you find these are spaghetti plots of these individual gene sets, and you can see the aberrant gene sets and where they're actually, what cells are driving the aberrant expression of these. And so this led to a bigger study where we were looking at, and I don't want to call them pseudo time series data because it was actually 4 individual mice, but we were able to stitch these cell differentiation vectors together across time. So Ash, we're running out of time here. I want to get the rest of these questions. But what you see here, and I don't have time to go into the experimental design here, but it was an ApoE network, knockout network, and then we had an ApoE and the TGF-beta knockout the receptor knockout that I had showed you earlier. That started this single cell initiative. As you're moving out radially across both of these directed acyclic graphs here, I want to draw your attention to the double knockout. So that signal is much stronger than the double knockout than the ApoE knockout. And as you're moving out radially, you're moving across time. You're looking at when that tissue was harvested in different animals. So it was a 4-month experimental design. You have a virtual ancestor here where we believe that these cells are fully differentiated. And then we are tracking these cell differentiation vectors across time until you get to this blossom here at 3 o'clock in the double knockout. And the beauty of these networks is that these were smooth muscle cells. And we could track the expression of markers of known smooth muscle cell markers. So this is myosin heavy chain 11. You see that you have the loss of expression. Can you go to the next slide, Ash? And in these same vectors, can you go to the next one there, Ash? There you go. And in the next, where you see the loss of expression of what, a marker for smooth muscle cells, now you're seeing the expression of inflammatory signals. So we knew that we were hitting the right disease vectors. And if we go finally here's the phenotypic support for this. You've got a cross section of an aorta here. Mike Simons was able to capture an aberrant population of smooth muscle cells clawing its way in through the endothelium that actually led to disruption in aortic aneurysm. And where we're looking at it now, these signals are so specific with the differentiation signals. Here is the abdominal or the ascending aorta, which I just showed you, and then the abdominal side of things, aneurysms here. We found that same vector, but we found 2 additional vectors as well. So the answer to your question is yes, we can do that at the single cell level. Now with spatial transcriptomics, it's like supercharging these analyses. So we did not have access to spatial transcriptomics at the time. So I hope that answers. That was very, very long-winded, but I hope that answers your question.
Okay. We have like 10 more questions. So I think, and we're also we're at about time now. I think maybe we can answer one more and then call it. So let's see here. Here's one. Paste it in here. What spatial transcriptomics platform are you using, and how do you deal with the challenge that the abundance of transcripts captured can vary from sample to sample? Can the results from one ST dataset be generalized to others?
Yes. Yes. Very, very, another good question. So it's 10x Visium that we're using with this. I seem to like it better than NanoString, and there's a number of different reasons for that. But your question here is deal with the challenge of the abundance of the transcripts captured. Yes. So do you have to get into imputing data? I've never liked that. You have much more than 10% of the overall signal on these transcripts across. And can you go clear back to slide? Yeah, what slide number is that? The very early ones with the digital pathology that I opened up. Yep. Nope. Here, let me drive here. Sorry. Now I've lost it. Okay, there it is. Okay, so you've got this tissue signal. You got 6,000 spots, and this is a problem that's plaguing spatial transcriptomics. But the way that we're filtering that, we actually come up with a pool of signals. In some cases, it's analogous to what we've dealt with in the past with single cell and dropout. We can fit zero inflated approaches or data distributions and add the zero inflated negative binomial approaches with our single cell approach. You can impute data as long as there's not more than 10% of the overall signal. From a statistics standpoint, I'm okay with that. But when you get a lot of dropout or missing signal, this is still problematic. So we use a combination. I hope this doesn't sound like I'm skirting your question. It's a combination of looking at the data, the specific experimental design. Do we feel comfortable about imputing data, or do we filter the data based on a point that you have a common pool of signals across all of these spots? Do we filter out the spots that don't have an adequate amount of signal? That seems to take care a lot of that sparsity that plagues both single cell and spatial transcriptomics. And I think there's probably another aspect to that question that I haven't addressed here. Can the results from ST spatial transcriptomics? Yes. So generalizable. So that is where all of the analytics come into play with that, is that if we're capturing enough biological variation and we put it together within a spatial or within a structural causal model, that is very, very generalizable. So if we move away from what's correlating or with a disease state versus what's driving it, what correlates versus, what's actually driving it, then that speaks to generalizability. It's not perfect, but it's much, much better than what we have found in the past.
All right, I think we're at time now. Thank you so much, Dr. Chinden. This has been amazing. We also received so much engagement and participation, which is really exciting to see. I posted in the chat here, but we will answer the remaining questions in a follow-up email. So I do apologize if we weren't able to get to everyone's questions. There were many. But again, thank you so much for carving out time out of your busy days to join us today. You'll also all be receiving a recording of the session automatically. So again, thank you.
Thank you.